Welcome back to this course on organic chemistry in biology and drug development. In the last session, we have discussed the process of translation, and before that we studied the process of transcription. Again, just to have a quick glance of the information flow in biological system or flow of information, that is the DNA is first copied, self copied, and that is a semi conservative replication. That process is basically from double strand DNA you get a copy DNA, and then the DNA has to be transcribed that to a messenger RNA and also the other RNAs that are involved in the subsequent steps, that is translation. So, transcription is the process of copying of the DNA into the RNA, and then from the RNA you by the process of translation the proteins according to the sequence, that is, or according to the codon that is inscribed in the RNA, that is the messenger RNA, particular type of RNA, and that gives the primary structure of the protein that will be synthesized. Okay. So, replication, transcription and translation are the three fundamental processes of living system. If any one of these can be stopped, then the cell division, that is mitosis, that cannot take place. So, the cell will die automatically. Now, just a quick glance of the transcription and translation. Remember, in replication always try to find out what are the enzymes, because we will always look at the organic chemistry perspective of the processes that are involved in replication. Now, in replication there are a lot of enzymes or proteins also which are not having enzymatic activity, but binding affinity, they are involved. Now, if I start to name all these enzymes, you know, the first thing is you have to break the helix. So, a helicase enzyme is involved. Then you have to have DNA polymerase that will synthesize the or bring the building blocks and synthesize the polymer. Then you have the primase which makes the primer, which are made up of RNA. So, primase is basically an RNA polymerase which makes short primer sequences made up of ribonucleotides only. And then you have the generation of the Okazaki fragment. We have discussed that there is a problem in going from the 3' to 5' direction, because the DNA synthesis takes place from 5' to 3'. So, it just it is a discontinuous process. You get fragments which are called Okazaki fragments. So, you need an enzyme which is first of all an endonuclease which takes care of the RNA primers, chops it off, and then that is replaced by the proper oligonucleotides, okay, proper polymeric, short polymeric oligonucleotides. And what is left after that? It is that ligation process, that you have to join these pieces, Okazaki fragments, and then via an enzyme which is called DNA ligase. Okay. Besides that, there is this SSB proteins, that is single strand binding proteins, which whose job is to keep the two strands apart as long as your DNA polymerase is working to make the double strand. So, it is there are so many enzymes that are involved, and so many proteins, non-enzymatic proteins are also involved, like SSB proteins. Okay. We will just have a brief glance of try to see the mechanism of this, but one more enzyme I forgot to mention, and that is that when you unwind the DNA, there is a strain that develops in the front of the replication fork, and that is supercoiling. So, in order to reduce that whatever stress that is generated there in the ahead of the replication fork, so that has to be taken care of, and that is taken care of by an enzyme called DNA topoisomerase, because it is basically a topological problem that when you disturb at some location of a system, if it also disturbs the geometrical properties ahead of it or somewhere else behind it, then that is called a topological problem. So, you have a topological problem which is generated by supercoiling, and that has to be reduced, taken care of, and that is done by the enzyme which is called topoisomerase. Okay. Now, in the replication, that is the replication, and in the transcription we have the I told you in transcription what happens, that there is this, first of all, there is a promoter region where the RNA polymerase binds at the promoter region, and I told you about different promoter regions. One is the Pribnow box. In prokaryotes, we are talking about prokaryotes, and then prokaryotes you have the -10 box, which is called the Pribnow box, and that sequence is from 3 prime to 5 prime. If you read, that is TA, TA, AT, okay, that is the and G, but this is not followed all the time. If it there are some variations, or there is some statistical analysis that how many percentage of the bacterial population really has this type of sequence, but this is the sequence that was obtained from particular type of bacterial strain, and if this sequence is present as the Pribnow box, then the attachment of the RNA polymerase with the strands of the DNA. Remember, there are again two strands, one is the coding strand, another is the non-coding strand, or one is sense strand, another is one is sense, another is antisense, or you can call it you can also call it a template strand and a non-template strand. Okay. So, the binding to the strand, to the template strand will be very high if this sequence is there. Perfect homology is there in the bacteria. Okay, that is the Pribnow box. Then you have the -35 box. Remember, this is actually the upstream. So, if you have the first base of the RNA, that is considered to be the +1, which is transcribed, then we are talking about on this upstream, that means -35, that means it is on the, if you write it from the 5 prime to 3 prime. So, it will be on the left side, and that is called the -35 box, and there is again a consensus sequence, but again I remind you that it may not be perfectly followed by all bacteria. Okay. So, that after the binding of this, and there is this RNA polymerase is a pentameric protein, and then there is a sigma factor, and that sigma factor is the one which is actually binding, recognizes this sequence, and then binds to the DNA strand, and then the sigma factor falls off, and the RNA polymerase moves downstream and bringing one base or the other. Okay. And in the, so this is again the same thing, and in, as again I just repeat what is the, this is your sense strand or DNA coding strand, non-template strand. These three are the names that is given to this, and this is basically, this is the DNA template or antisense. Sometimes it is also positive strand or negative strand, that is also another terminology, or it can be called non-template strand. So, you have to be very careful when we say something, then you just have to be very quick. So, all these different names for the same strand are possible, and different books will utilize different names. Okay. So, here the antisense strand is this and this is the sense strand. Why it is sense? Because this is the sequence which will be transcribed ultimately in the RNA. The RNA instead only difference is there are two differences. One is that T is not there in RNA, it will be U, and the other is it is ribose instead of deoxyribose. Okay. So, I think these are all done last time. So, this is the process, that is the sigma factor, you see this is, that means the PRIB now box and then it falls off. So, now this sigma factor is not there. So, the RNA polymerase can move and bringing in the oligonucleotide bases, okay. And then oligonucleotides, not bases, oligonucleotides, and then the subsequent formation takes place. Remember, this synthesis also goes from the 5 prime to 3 prime direction, and once it is done, once it reaches the termination step, then the everything falls off. Okay. I also told you about the so these are the different steps: template recognition, RNA polymerase binds to DNA, DNA unwound, initiation, elongation, and then termination. Okay. Here it is just initiation, and then basically the sigma factor is detached, okay, is detached, and then it can move and synthesize the complete RNA. Termination process is basically, it has a particular type of sequence or particular type of geometry. As it reaches, then it falls off at that time. So, there are two ways to do that. One is that I told you that last time, that there is a GC rich region, G and C rich region, which forms a hairpin, and once the hairpin, after the hairpin, it is basically poly U. There are so many U's are there, and these U's are basically the U, you know that U complements with the A, but that is the only 2 hydrogen bond forming process, and then so that means that part will be very loose. So, that can easily melt, that means the separation can be easy. So, the as it melts, it falls off from the DNA and then the whole RNA comes out. The other is that there is a row dependent process, which I also told you. Row is a protein, and this row dependent process utilizes ATP, utilizes the energy from the ATP, it hydrolyzes, takes the energy, and then it releases the, in the process, the RNA, because the energy is utilized in releasing the RNA from the nascent RNA from the DNA strand, from the template DNA strand. Okay. So, these are the different processes. Now, let us so in the replication, we have DNA, the enzymes, that is DNA, first of all DNA, that is helicase. So, we are talking about DNA to helicase. Okay. So, DNA to DNA, helicase, ligase, polymerase. There are 2 types of polymerase, means one is DNA polymerase, another is RNA polymerase, primase, and then you have topoisomerase, okay. And in DNA to RNA, that means the process of transcription, the enzymes that are involved is polymerase, RNA polymerase, and you have this other things, RNA polymerase. This is little bit simpler. You have the RNA polymerase, which is actually having a pentameric protein. So, you should know about what is a sigma factor, then what is a rho factor, okay. So, usually these are the ones, rho factor, and the mechanism of. So, here the important point is the initiation, that how does it initiate and how does it terminate. These are the important questions that we have answered. And in translation, translation means RNA going to the I think this is the same thing, the helicase. I just told you what are the different, that is shown here, what are the different enzymes that are required to do the replication. And let us talk about the translation. We have not talked about the translation, what are required in translation. Translation is the process in which you now make the RNA, the mRNA to protein. Okay. So, that is called translation, and here the enzymes that are involved are, one is that amino acyl, synthesis of amino acyl tRNA. Okay. Remember, this is the enzyme which incorporates the amino acid as the ester via the ester linkage, and it is hooked to the 3 prime overhang. Overhang means you have this is the more or less the geometry of the RNA. So, this is the 3 prime end and this is the 5 prime. Okay. So, this overhang means that you have an extra which is not connected with each other. Earlier there was definitely this type of complementarity is there. Okay. And this is, there are 3 bases, which are called anticodon. Okay. So, what happens? This 3 prime wedge is attached to an amino acid via an ester bond, that means, it is hooked to the carboxylic functionality of the amino acid. And there are how many, if the question comes, how many tRNA are involved in amino acid in peptide synthesis, then the minimum number has to be 20, because there are 20 amino acids that has to be brought in to the ribosome, where which is called the protein making factory, and that so that means you need 20, because 20 tRNAs are there at least, but there are more, because you know this anticodon, because the codons are, some of the codons are can be degenerate, that means, there are number of codons which codes for the same amino acid. So, you can have these anticodons, that means, there are 64 anticodons possible here. Okay. 64 anticodons possible, and some of the anticodons signal the also the stop. They do not bring any. If the anticodon happen to be stop codon, that means, there is no amino acid attached to the 3 prime end. Okay. So, the one enzyme which incorporates this amino acid to the 3 prime end, that is the amino acyl tRNA synthetase, okay, because that makes the OCO, then the R and NH2. Okay. So, that is one enzyme, very important. Number 2 is basically the formation of the peptide bond, Okay. And you know what is the mechanism of that? I told you that here, just briefly describe that when the mRNA first binds to a region which is present in the rRNA, ribosomal RNA. So, it binds, recognize. So, there is a recognition sequence in the ribosomal RNA. The messenger RNA comes, recognizes that, and then sits in the ribosomes have 2 subunits. I told you about these subunits, a small subunit and a large subunit. So, so that mRNA, which is now sitting over the it recognizes the sequence, or that recognition sequence in the rRNA, and then it spreads along the ribosome. Okay. And then there are 3 sites in the ribosome. I told you, one is called the exit site, another is the P site, and the third one is A site. A is the amino acid site, P is the peptide site, and E is the exit site. So, what happens there, that the first remember, the starting point of the peptide synthesis, the starting amino acid is methionine. Unfortunately, methionine has only one codon, and that is AUG, starting from 5 prime to 3 prime, AUG. So, then methionine, the methionine containing the tRNA, that will bind to the P site. Okay. And then in the A site, the next amino acid which is required for the protein, and as dictated by the codon, next codon, that amino acid tRNA will bind. Okay. So, very simply we can have these 3 sites. This is the exit, this is the peptide site, and this is the amino acid site. So, initially this is the tRNA containing methionine MET, and this is the containing the amino acid which is dictated by the whatever is the codon sequence. Okay. Now, there is a reaction between this, and the methionine is transferred here. So, the peptide that is made is a dipeptide, methionine, then CONH, and then this amino acid, okay, dipeptide. Then what happens? The polymerase, the ribosome moves, and then what happens as it moves, then this tRNA, which is now devoid of any amino acid, because the methionine is transferred here. So, that goes to the exit site, and finally, it goes away from the system. And what happens now, this peptide, as it shifts one frame, so then this whatever is attached here, that will come in this site, and this will be free. And here the codon which is present here in the mRNA. So, depending on that, the corresponding tRNA will bring the next amino acid, and then the tripeptide will be formed, and this process follows till a stop codon comes, and then everything is released, the peptide bond is released. Now, this is a reaction which is the peptide bond formation. So, the formation of peptide is catalyzed by, interestingly, is catalyzed by the RNA itself. Okay. See, there are, I told you that splicing, that intron exon thing, when the for eukaryotes, the RNA is formed, the messenger RNA, which is first biosynthesized, that is formed, which is called the immature or pre-mRNA. Pre-mRNA means when there is this intron and exon, both are present one after another. And then what happens? Then there is the enzyme which is called spliceosome. Spliceosome is basically the RNA itself, and it takes care of the intron, cuts the intron away, and joins the exons one after another. So, takes the intron away and joins the exons one. So, exon 1, then intron 1, then exon 2, then intron 2. So, after the reaction, what will happen? Exon 1 join to exon 2, then join to exon 3. Okay. So, that is catalyzed by the RNA itself, okay. And that is a very remarkable discovery, because that that breaks down some of the earlier concepts, that enzyme like activity can only be shown by or biocatalysts are only consisting of proteins, which is not true, because there are RNA molecules which can also catalyze reactions. These are called ribozymes. Okay. So, now let us see how these processes, some of the enzyme processes occur. We will discuss the mechanism in as simple terms as possible. Okay. Remember, I told you that when the replication fork moves, then there is a stress that is generated ahead of the replication fork. Okay. And so, when there is stress, that means the corresponding the strain will be involved, or the strain will be generated, and then to release that stress or strain, then you have to, what happens? You have to then unwind, means you have to rotate the DNA, because there is excessive supercoiling ahead of the replication fork. So, you have to release that stress, and that is done by the DNA topoisomerase. Okay. The way it is done, I can show you actually how it develops, that if you have let us see, if you have a DNA which is, we know that it is a right handed double helix, it is a right handed system, right handed helical system. So, what we are saying is that okay. So, this is the system, if you can see it, it has got a right handed turn, right handed helical pattern. Now, what we are saying that if you suppose I fix it on this side and then you try to unwind the system. So, what happens, the helix, earlier the helix was spread out from here to there to my this hand, and now as it unwinds. So, you see the helix are basically now the number of helix is spread out in a very short region, and that is basically what is the supercoiling. And as you move it further, it is very difficult to move it, because it gives a, I said that it creates a stress ahead of the replication fork. Okay. Now, how to release this stress? You just cut one of the strand and then turn it from the, it is actually the strand which is above, you cut it and then you bring it from at the bottom point and then reseal it. Okay. So, that or reseal, or you just hold it for the moment till the replication fork almost is near to this end. That means, the DNA topoisomerase works by what is called a nick, by nicking or producing a nick in one strand, and that helps the strand to rotate and then rotate in the opposite direction and release the stress that is generated. Okay. And as the DNA synthesis is going on, the polymerase works. So, as it comes near to that point, the nicking is again sealed, and the nicking has to be sealed, because you have to have the DNA polymerase will move forward. So, there cannot be any nick. So, nick has to be sealed, and the DNA topoisomerase goes further ahead of the replication fork, and then again there is this supercoiling that happens there. So, it does again nicking there and then release the stress. Okay. So, that is how the DNA polymerase works. The question is that how these nicks are formed. Okay. Now, DNA polymerases are different, these topoisomerases can be of different types. It can be topoisomerase 1, 2, 3, 4. We are not going into that details. We are just showing one mechanism by which the nicking of one strand is carried out. Okay. Remember, nicking means you are breaking a particular strand. Okay. And how do you do that? What is the, so this is the DNA strand. Okay. See, this is the 5 prime phosphate, and this is attached to other sugar base, those one, and this is the phosphodiester, and this is the, suppose this is the phosphodiester, and this is the next DNA, the sugar base system. Okay. Now, what happens, basically we are saying that the strand has to be cleaved. So, these are the double strand, one strand needs a nick. Okay. So, that means there is a, it is not joined, okay, like this. So, once this nicked, so this can turn around and release the strain. So, this nicking is carried out by just hydrolyzing the phosphodiester bond. So, if you hydrolyze the phosphodiester bond, then there is this no joining between the two ends here. This is the 5 prime end and this is the 3 prime end. So, then how it is done? It is in topoisomerase 2, in the active site there is a tyrosine. Remember what is tyrosine? Tyrosine is nothing but an aromatic amino acid with a para-hydroxy group. Okay. That is a phenolic wedge, and this wedge can be a good nucleophile, wedges are good nucleophile. So, this attacks the phosphorus. So, it is a phosphodiesterase basically, and then breaks this bond towards the 5 prime end. This is the 5 prime end. Okay. So, that breaks. So, this becomes OH and this is the phosphodiester. So, a transient phosphor, not very transient, means it is an intermediate, I can say. It is momentarily formed between the tyrosine and the sugar, the next sugar phosphate backbone. Okay. But your job is done, there is a nicking now, no joining between this part and the other part. So, now the turning takes place and that stress is released and the fork can proceed further. Okay. And then when you require the religation, that means this is cleavage and you require the religation, then this wedge will come, attacks this phosphate and the tyrosine is released. Okay. So, it is a reversible reaction. So, via tyrosine you can do this phosphodiester cleavage. So, that is one of the mechanisms of the topoisomerase, particularly this is topoisomerase 2. So, it is a tyrosine mediated phosphodiesterase linkage. So, here that is what is shown in the topoisomerase, that basically what happens, that as the replication fork moves, then you have this supercoiling. So, topoisomerase is like a scissor and the scissor cuts one of the strand and then the strand rotates and then there is what you see is that a bubble, a replication bubble, but which is now not attached by the base pairs. Okay. That means, if you have n number of base pairs from here to there, so you now will have n minus 1 base n, not base pair, sorry, this is the turn. So, you have n turns here. You remember what is the turn? Turn is a complete turn is this, and so you have one less turn in the duplex DNA. Less turn means less coiling. Okay. So, you have to produce a n minus 1 turns out of the n turn duplex DNA. That is what exactly this is done. Let us talk about the spliceosome. That is, I told you what is spliceosome. Spliceosome takes care of the intron problem. The pre-mRNA or the immature mRNA goes to the matured mRNA. Okay. Now, what happens here, and the RNA itself is your enzyme. Okay. This is the catalyst. I should not call enzyme. The RNA itself is the catalyst. Okay. What has been found, that intron, there is somewhere there is a means the adenine. Okay. So, now this is, remember, these are not deoxy systems, these are only the normal ribose. So, the A will have the 2 prime OH. So, what was found, this 2 prime OH attacks this phosphate linkage here, which is attached to the last base of this exon. Suppose this is exon 1, this is your exon 2 and this is intron 1. So, this A is residing in the intron and the 2 prime OH is attacking the phosphate linkage between the terminal base of the exon 1 connected to the first base of the base sugar of your intron, and that phosphodiester linkage is cleaved. So, what will happen now? You will get a system where this 3 prime OH will be free. So, what we are saying is that this is the exon. So, you have OPO, O minus, O, and then this is the intron. Okay. So, there is a here in the intron and that A has a 2 prime OH and this attacks that one and releases this one. So, you get a 3 prime OH, this is the 3 prime and 3 prime OH is free, and in the process you get this circular type intron, but it has got some attachment here to the 3 prime end. Then in the second step, this is hydrolyzed and this is hydrolyzed means this is not hydrolyzed, this is sorry, this is now the 3 prime OH which is generated, very interesting mechanism, that acts as a nucleophile and cleaves the phosphodiester linkage here. So, this is O, PO, O and this is O minus. So, now what will happen, the OH which is generated here, that attacks this phosphate and releases the intron. So, the intron is released and the exon 1 and exon 2 are joined. So, that is one of the mechanisms of the spliceosome. Okay, that's an internal, it's basically internally done. Okay. The 2' OH. Now, you can say why the 2' OH is a nucleophile here. Usually what is found that the bases that are present in the RNA, they are like say adenine or guanine. They have this imidazole ring, okay, imidazole attached to a pyrimidine ring, fused to a pyrimidine ring, and these are quite basic. So, it can be that imidazole base is the one which activates the, we should call that purine bases. The purine bases activates the 2' OH by abstracting this hydrogen. Okay. And then when this hydrogen is required to be delivered, it delivers it. So, that is what is the mechanism of spliceosome catalysis. Okay. Then the formation of this amino acyl tRNA, that is done by see, this is the amino acid and this is your ATP molecule, ATP triphosphate. So, the first this is activated, the carboxylic end is activated as the phosphate. So, first this is AMP, amino acyl AMP, because you have a monophosphate now. So, now this 3' OH attacks the, this is activated now, attacks the carbonyl end of the carboxyl ester, and then, so basically this is a mixed anhydride. One is the phosphoric acid, another is the amino acid, and this mixed anhydride is a very activated system. So, the 3' OH attacks this carbonyl and releases the AMP. So, AMP is released, and what you get, that this is transferred to the 3' OH, okay. And this is the tRNA containing the amino acyl group. Okay. So, this is basically the tRNA amino acyl synthetase is it is called synthetase because it requires an ATP to do the, to catalyze the reaction. Remember, there are two types of you can also categorize two types of enzyme systems. One is synthetase, another is synthase. Synthetase. So, synthetase is the one, both are actually synthesizing something. Okay. If it is synthesizing X, then it will be called an X, synthesis of X. So, the enzyme you can just abbreviate it quickly as that X synthetase or synthase. Now, both are synthesizing, the difference is the synthetase utilizes ATP as a cofactor, and then that will be called synthetase. If the synthesis does not require ATP, involvement of ATP, that will be called a synthase. Okay. Now, this is the mechanism of the peptide formation. Now, the translation, there is this, first there is a methionine, okay, and that is attached to the methionine, you know, that is the R group, the NH2 is free, and this will be attached to the tRNA, okay, tRNA, and the tRNA contains an anticodon which is specific for, which is the opposite to the codon required for expression of methionine, and the other tRNA has also that is attached via the 3' group to this. So, something say alanine, the second one, and this is the NH2, and the reaction that takes place is this attacks the carbonyl and that goes out. So, the tRNA containing the methionine is free, and this is transferred to the next one. Okay, I write here. So, now you will have a tRNA which is attached to your alanine and then methionine. and then as I said, there is a movement of the shift of the polymerase. So, this goes to the P site and the A site remains free, but depending on the codon, now the next tRNA will be brought. Now, who catalyzes this reaction? This is catalyzed by ribozyme. Ribozyme catalyzes this reaction simply by, again very similar, that if you have a pyrimidine base, you have lot of basic nitrogens, one is this one, another is that one. In this case, depending on the proximity, it is not dependent. Remember, in enzyme catalysis or biocatalytic systems, it is not always the most nucleophilic or most basic nitrogen that is going to assist the catalysis. It is the proximity effect. So, the way it is done is that this nitrogen is proximal to the see what happens here, to the initially this goes on this side, okay, as the nitrogen attacks. So, that becomes a tetrahedral intermediate, and this O minus, that intermediate, tetrahedral intermediate, is stabilized by the protonated form of protonated form of the adenine base. So, that will now stabilize. So, it now assists in generation of the tetrahedral intermediate, which is required for the peptide synthesis. So, that is this is provided, this base is provided by the RNA itself, and that is why this is called a ribozyme. Ribozyme is RNA as enzyme are called ribozyme. That is a very general terms, but ribozyme also includes, remember, ribozymes also include the spliceosomes, which takes care of the intron. Okay. I think we have just have a quick glance of the or recollection of whatever was taught. I just again because it is a complicated area, replication, transcription and translation. So many enzymes, proteins are involved and so many mechanisms are there. That is why I thought that before going to the next topic, we will have a revisit of those topics. So, that has been done now. So, in the next session, we will go to our next topic, that is the amplification of DNA. Okay. Thank you.